bacteria forms the endospore. Not all 100% bacteria forms the endospore. Only 3 to 4 genera of the bacteria are able to form the endospore into the cell. Why these are called as the endospore? These are formed within the cell, and after the formation, these are get released out of the cell. And these, what are these adverse environmental conditions? Or, for example, heat, dehydration, radiation, and presence of the strong chemicals into the environment, like strong acid or strong alkali. Then, if these conditions are present into the environment, then bacteria forms the endospore, and these endospores are dormant structures. There is no metabolic activity into the endospores takes place. Only these are resistant to the heat, dehydration, radiation, and resistant to the strong chemicals. These endospores are structurally more and more complex as compared to the vegetative cell. Because of the complex nature of the endospore or complex structure of the endospore, these are uh, resistant to the various adverse environmental conditions. Okay, this endospore, if you see the structure of the endospore, you are going to study it in your theory course. It is very complex structure, multi-layer structure, and hence it is resistant. The two species of the bacteria, this endospore are present. Okay? The endospore staining we can do with the help of the specific method. Okay, so heating is used as the accentuator. Accentuators you might have been studied in first term. These accentuators are held to penetrate the stain inside the biological specimen. It may be the spore or vegetative cell. Hence, heat as the physical accentuator, it is used in this Schiffer and Fulton staining. It is also used in acid-fast staining to stain the acid-fast bacteria. This heat, it is used here into the endospore staining. There is no need of the decolorization of the spores once they are get stained with the malachite green. So they will not uh, uh, leave this stain once they got the stain with this malachite green. So in fact, even though we washed with water, these are not going to wash, but the vegetative cells are get washed. Okay? So water, it is used to decolorize the vegetative cell or to wash the cells. These endospores are get stained with the malachite green, that is a green in color, as the primary stain used into the endospore staining. And into the counter staining, if we added the saponin, the vegetative cells are get stained with the saponin, and this appears the red in color. So in details, we will see this method of the endospore staining. What will require? What are the requirements for this staining? Slide, wire loop, spore forming bacteria, then primary stain, malachite green, then counter stain, saponin, then boiling water bath, okay, to generate the steam so that we can heat the smear and we can apply the accentuator, then microscope to observe the this slide under the oil immersion lens, and then oil immersion lens, cedar wood oil for observation of this stained smear. Okay. Then procedure. What is the procedure? Prepare the smear of the given organism. Okay. You take the clean grease-free slide shown here in this picture. Then after that, take the loopful suspension of this test organism or given organism and make the smear and allow to air dry. Okay. Second step is the heat fixing of the smear. Take the slide and pass it through the flame 2 to 3 times so that that smear will get fixed. Okay, so heat fixing of the smear is very important, and I already told you why heat fixing is important, because heat fixing is important to heat the cells, and cells are made up of the proteins, and coagulation of the proteins takes place and they sticks onto the surface of the glass, and so that even though during the staining procedures there are the washing steps, so the cells remains attached onto the surface of the slide. These cells are not going to wash during the washing step. Hence the heat fixing of the smear is very important. Hit the slide gently till it starts to evaporate. Okay, so that after flooding the stain we have to heat this slide, okay, so that the stain will start to evaporate. So here heating will apply as the accentuator, and it will increase the porosity of the spores, and this stain will get enter into the stains. Okay, this staining you can see here, the after preparation of the smear, the slide, it is a smear, it is flooded with the malachite green, and after that that slide, it is placed on boiling water bath. It will heat the smear so that it will get heated, and that heat will act as the accentuator for the staining of this cell. Add the malachite green and see whether there is steam is coming out from the smear or not, and just observe the steam evaporation of the uh, this stain from the smear, then again add it. So this you have to do for 3 to 5 minutes. In this way you have to do for the 3 to 5 minutes. After 3 to 5 minutes, take the slide, okay, take the slide and wash it under gentle flow of the tap water. Wash it under the gentle flow of the tap water. So that you will see here in this slide, in first step, while applying the primary stain malachite green, all the spores as well as the vegetative cells here, these are get stained green in color. But during the washing, the this green color vegetative cells are get colorless, but not the endospore, because endospore once get the stain with the primary stain or malachite green, they the endospore never loses the stain, and they appears green in color. And after washing, flood the smear with the counter stain saffronin for 1 to 2 minutes, and after 1 to 2 minutes, wash the slide again under gentle flow of the water, and then observe the smear under oil immersion lens under the 100x. Okay, so you can see here what happens during the staining. These cells will get stain as well as the spore will get stain, okay, while applying the malachite green. But washing, during the washing, this vegetative cells will lose the this stain. Your stain will get easily removed from the vegetative cell, but not from the pores. And here, by applying the counter stain saffronin, this vegetative cell becomes the red in color and spore remains green in color. So in this way, we can stain the endospore green in color. And we can observe if this green color structure presents within the cell or presents into the smear, we can say that that bacteria are able to form the endospore. And if it is not present, then we can say that the bacteria cannot form the endospore. So in this smear, you can see in this slide, there are the red color cells and green color endospore. Are you getting it? So So these are the green color endospore and red color cell. So this was